Welcome everyone to today's session. My name is Conor Moriarty and I'm Head of Network Strategy and Operations at Diagnostics. We have a look at our agenda for today. I will introduce Diagnostics, our company, first of all, explaining a little of who we are. I'll then have the privilege of introducing our specialist speaker for today, Professor Sarah Ennis from the University of Southampton. Very excited to hear and learn from her. We'll then have some questions and answers at the end. So to begin, a quick word on Diagnostics, our business. Our roots are in consulting to the pharma industry with a particular interest in precision medicine. We believe that through leveraging of data and technology, we can enable better testing, better treatment, and ultimately provide more life for patients. It's powered, our use of this technology is powered by DXRX. It's our platform which really drives this approach using data and technology, increasingly incorporating AI for things like error correcting in testing data and predictive analytics. So we're delighted to be hosting this topic today. But a note that through our journey over the last 20-ish years in business, we've learned that it's not the technology itself that really drives this. It's the integration and interaction of those benefits with existing industry and community scientific ecosystems. Those ecosystems are led by individuals like our attendees today. So we're honored to have you with us. If you'd like to hear more or join us on that journey, our DXRX network is free to join. It acts as a central hub for information, collaboration opportunities, and the provision of events like this one today for our members. So we'd love to see you over at dxrx.io sometime in the future. Now, it's my tremendous privilege to introduce Professor Sarah Ennis. Sarah has told me that she's been working with large-scale genomics data since the last century, just to make everyone feel nice and young. And she currently runs a diverse team who leverage high-performance compute resources as their laboratory to really uncover insights and innovation in this sector. And she'll talk us through a lot of her insights and knowledge during the presentation today. And before I hand over the floor to Sarah, I'd just like to express our gratitude on behalf of all the attendees today, on behalf of Diagnostics and LabRoots for her giving her time and sharing her knowledge with us. Sarah.
Thanks very much, Conor. That's a really nice introduction. So it's a privilege to be here talking today and to be starting to share some of what I believe is we're really only scratching the surface on the potential of the use of large-scale or deployment of large-scale algorithms and AI to make genomic data work better for patients. So I think, as Conor mentioned, I work for the University of Southampton, have done for many years. And so a lot of the results I present today come from some of the work I'm doing in that dry informatics laboratory. But also I work 1 day a week in the genomic medicine service within the NHS. And so I think the reason I mention that is the unifying thing about whichever hat I'm wearing is that I'm motivated by integrating high throughput genomic data with rich, accurate, reliable, standardized clinical data for the purposes of making the data work harder for patient benefit. So that's my raison d'etre in my job. Genetic data is fascinating, but we've got to make it work. It's expensive. We've got to make it work harder for patients. We've got to diagnose more patients, get more patients back to school, back to work, prevent more disease. And that's the common theme, whether I'm wearing my academic hat at the university or my research director hat within the NHS. And as you can see from these NHS long-term plan and a lot of the Genome UK guidelines from the UK government, these are common themes that sing from a similar hymn sheet in their literature as well. So given that context, I'm going to split today's talk up into two main things, because it is all about those, well, really three things. Analyzing genetic data for patient benefit means that you have two sorts of data: the genetic data and the high integrity clinical data. And sometimes that may be longitudinal data as well. But you need that high integrity, rich, structured clinical data, and you need the genetic data that we're increasingly able to produce at vast scales these days. And so initially I'm going to talk about those genetic data and some tools we're using to compress them, because of course the problem is it's quite easy to extract a DNA sample these days, put it on a machine and generate terabytes and terabytes of data. But how do we handle those data? How do we handle the ever-growing sparsity that is genomics data? So I'll talk about that and a tool that my group have developed to deal with those data called GenePy. And then I'm going to switch tack and talk about clinical data. So in my role in the university, I'm also chief investigator of some studies that actually directly recruit patients from hospitals. And I have a cohort of patients diagnosed with inflammatory bowel disease. So very common disease affecting an autoimmune disease, affecting any part of the gastrointestinal tract. And I have about 1,500 patients for whom I've got these genetic data and rich clinical data. And I'm going to demonstrate the value of bringing these two data types together for those patients. So moving firstly to GenePy. So this is a tool that I developed because I started sequencing those patients in about, recruiting them in about 2010. And of course, 2010 was just after the real fury of effort towards genome-wide association studies. So throughout the sort of early years after the human genome was sequenced in sort of 2004, 5, 6, 7, there was a hotbed of research globally applying genome-wide association studies. And they were great. Up to that point, we had largely been successful really in only identifying the genes behind single gene disorders. So LOD scores were the method that was used to analyze how DNA segregated between generations in families and identify single genes like the cystic fibrosis gene that would underpin individual diseases. And it was really when we moved into genome-wide association studies that we started to unpick some of the genes that underpinned the more common diseases that unfortunately many of us end up getting diagnosed with. So whether we're talking about Alzheimer's, Parkinson's, psoriasis, asthma, chronic obstructive pulmonary disease, psoriasis, all of these common diseases, GWAS did a great job at identifying some very commonly genes that would commonly be impacting and influencing the susceptibility and the outcomes to these diseases in individuals. But there was a problem. The GWAS would identify new pathways and important genes, but in actual fact, if you had a patient with psoriasis in front of you, it was very hard to translate, well, what's the relevance of that gene that's been identified on a huge population? What is the relevance of that to this patient sitting in front of me? Are those data any good at all for that patient? And so I was collecting these IBD patients. GWAS had done a great job in IBD. We went from knowing one gene to knowing a lot of different genes that may influence susceptibility. But then when I would meet my consultant gastroenterologist colleagues in the hospital and I would say, oh, isn't genetics doing a great job identifying new disease genes that modify risk to Crohn's disease or ulcerative colitis? In actual fact, those data were not translating in the clinic. When this consultant was meeting his patients, he wasn't saying anything about these genes. It was not clinically translatable. And so there were a few things that were obvious. We were missing lots of the heritability. So even though we'd identified these key pathways that were important in, for example, IBD, the same thing happened in parallel across many other diseases. Even though we'd identified these genes, there was still the vast majority of the genetic component of the disease was not being captured. There was this estimate of missing heritability that was often up as high as 70 or 80%, that we weren't accounting for the observable genetics about 70 to 80% of the time. So that was one problem. So it had given us insight, but with a ceiling on the level of information and with a certain gap in clinical translation. And so this was about the time that next generation sequencing was coming about. So I decided not to start doing genome-wide association studies in my cohort, and instead I went straight to exome sequencing. That's sequencing the coding regions of all genes across our genome. So we're skipping all the intronic regions of genes, and we're skipping all those regions between genes, and we're focusing on that 1 to 2% of the genome that actually codes for protein. And it's estimated that about 85% of disease-causing variation lies within those coding regions. So it was the most efficient and economic way to get bang for your buck at that time. Whole genome sequencing data at that time was exorbitantly expensive. And even today, while it's much, much cheaper, the files generated in themselves are huge and very incongruous to handle and tricky to manipulate at scale, very expensive to manipulate at scale. So we've actually stuck with that, and we still to this day sequence the exomes of these individuals. And of course, when you sequence the coding regions of genes in individuals, what you start to uncover is not just the common variation, because the common variation is stuff that might occur, a variant that might occur at maybe at least 5 out of 100 individuals in any one group, or 5% of individuals, or maybe some people might interpret it as at least 1% or more. But in actual fact, the majority of variation that we observe in anybody's genome when we look at it, the frequency of it is much, much lower than that. And so it's actually rare variation that we observe, or even private variation. So that means it's variation specific to that individual, and you won't find it in dbSNP or other reference population data sets. And it's just specific to that individual or maybe their family. And that means that the traditional statistical methods we were using in genome-wide association studies don't work anymore. Because if all those individuals are private to, or all those variants are private to just a very, very few individuals, we have no power to observe differential frequencies in patients compared to controls, because only one patient has that new novel variant that you discovered when you got your sequencing data yesterday. So I was in the situation of needing some new tools to analyze our sequencing data on this IBD cohort. So as we sequenced more and more individuals, each time we would sequence another individual, we might observe some new small subset of additional variations not seen before. But each of those, as we sequence more and more people, are increasingly rare and rare. So the larger our cohort, the larger the space or the universe of variants we observe, but most of the variants are not present in most people. So it's a very, very sparse data set, and that's an attribute of all genomic data. It tends to be quite sparse. And so this was the issue. How do we deal with the sparsity of data? If I've observed some really nasty variant in a gene I think is important in disease, but nobody else with the disease has that variant, how do I interpret it? Or how do I collapse the data so that I can actually manipulate the data at scale? And that's how we ended up coming up with this method called GenePy. Now, this is a busy slide, and I realize I don't have a pointer. So if you manage to follow me through, if we start off with the little cartoon of individual 1 on the figure, on the top left part of the figure. So this is just the first person in any cohort, one individual. And of course, we've done the sequencing data, we've got the exome data, we've run it through a bioinformatic pipeline, and we've called what are the variants compared to the human genome reference sequence that we observe in individual 1 for gene A. And this individual has 3 variants in gene A. It may be if you sequenced me, I might have 7 variants, Connor might have 14 variants. And of course, they may or may not be overlapping. So how do you analyze those data? Well, for this individual 1 for gene A, we take each of those called variants and we annotate it with 3 things, annotate each variant with 3 things. a measure of how deleterious that variant is. And we use, and we have tried and tested many other deleteriousness metrics. We use CAD at the moment, but it is possible to swap that out for other metrics if people would choose. We use CAD 1.6 because it actually looks at variants, whether they're synonymous or non-synonymous variants. It looks at coding variants and non-coding variants that have just fall outside the coding regions into the introns. So we find it more informative for our purposes. We also look at the allele frequency of those variants. So if the allele is common and 50% of the general population has it, it's unlikely to be causing a nasty disease. And we use that frequency to downweight the value of that variant. And of course, for most genes in our genome, we've inherited a copy from mum and from dad. So we also incorporate the zygosity. So it's those three factors that we then use on each variant, and we pop those into the GenePy algorithm, which you can just see underneath the zygosity term there. And that creates a score for each variant. So for individual one, we sum up the scores for the three variants that are observed in their gene A, and they end up with a final score of 2.89 for gene A. And then you move to the second individual and create their score for gene A. You move through the entire cohort and you actually make an individual score for each gene in the genome. So in excess of 20,000 genes in the genome, we create this score for each individual. So it's a two-dimensional matrix, such as that you'll see in the bottom left figure, or the bottom left of the figure. So we have all the individuals on rows and all the genes in columns. And the genes or the scores are a quantitative score whereby the higher the score, the higher the burden of pathogenic variation in that individual for that gene. And it means that for any given gene of interest, we can ask who has the highest score, who has the highest loading of variation in that gene. And we can compare scores for individual genes between people. Between genes, the scores can actually change quite a lot because a gene like Titin or the collagen genes, for example, are huge and you can accrue quite a high score. And then some other genes could be very, very tiny and the maximum score might be much, much smaller. So then we sometimes do scale that score 0 to 1. But so we can use those are the data that we can then use in our downstream analysis. And we've gone from this massively sparse matrix whereby most people don't have most variations to this much more concise and limited matrix whereby you just have each gene in the genome, 20,000 columns by the number of individuals in your cohort. So that's the upper limit of the matrix then. Another way to actually use these data, so we can ask well, which individuals have high scores in which genes, but we can also, if you're interested in just looking at the individual level, you can in the first matrix on the bottom on the left, you can look at it an individual and you can move right away across the genome and you can ask, where does this individual have a high score compared to everybody else in the cohort? So it is that it's got that clinical translation because it's telling you this individual, for example, has a really high score in an epilepsy gene, much higher than the vast majority of the cohort. And lo and behold, they have a diagnosis of epilepsy. So maybe those two things are correlated and you may be just a faster step closer to making a diagnosis. But also, it might be possible to turn it on its head and look at it on a gene level. Maybe somebody has a particular interest in one of the collagen genes, and therefore it might be possible to rank all the individuals and say, who has a high weighting in my gene of interest? And what are the phenotypes? What are the clinical characteristics common to those individuals? So it's turning the old-fashioned paradigm on its head. You can have a genotype first approach whereby you're looking at, well, who has the massive burden of genetic pathogenicity in that gene and what is the phenotype that we find in them, rather than the reverse, which is where we're very often looking at, well, who has the disease and what genes are perturbed in the individual. So you can actually use this score going both directions. So I managed to press that button a little bit faster than intended. So the pipeline itself, I think it's just catching up. Yeah, the pipeline itself has a number of dependencies. We've published this on GitHub. It uses VEP, it uses CAD, as I've mentioned before. It's got the GenePy algorithm itself. It's got the massive amount of input data. It's possible to start from FASTQ files, or you can start also from VCF files, that's variant call files, so already processed genomic data. We use Human Genome Reference 38, and we use GenCode annotations. Of course, when you've got a very substantial pipeline like this, it can be ideal to wrap up some of the software in these containers, Docker containers, and then map those into a next flow pipeline so that that can be used, and the pipeline algorithm does not just work in our local high performance compute cluster. It's actually downloadable and can be run quite easily on different data sets in different centers. And that indeed is something that we're busy at the moment. So a colleague of mine, one of my postdocs, is working very heavily on generating these GenePy matrices within other environments. So we've got up here on the right-hand side, we've got the GenePy software and pipeline next flow all wrapped up on GitHub. We've already applied it to Iridis, which is our local high-performance compute at the University of Southampton. But on the right-hand side, we're also applying it in a Microsoft Azure cloud for the purposes of working with the secure data environments in the NHS. And on the left-hand side, we're working on Amazon Web Services environments, whereby we're deploying it within the Genomics England environment. And they have an instance, a LifeBit platform that sits within AWS, and we're installing GenePy in there. We're using it, obviously, therefore, on the Genomics England data set, and we're currently creating the GenePy matrix for 78,000 individuals for all genes in the genome. We're also in the process of applying this to the UK Biobank data, and that actually we're doing that both in the UK Biobank research portal, but we also have a local download of the UK Biobank phase 2 exome data, and we've already deployed that actually on our local HPC. And so I might mention some of those data later on. So that was 200,000 individuals for whom UK Biobank developed exome sequencing data, and we already have their GenePy matrix. And then we also have my local cohorts, these Southampton whole exome sequencing cohorts, comprising of patients, about 1,500 patients with inflammatory bowel disease, and another 500 or so patients I have exome data on rare disease. We have actually done a pilot analysis before we started deploying this across all genes for all patients in GEL. We did a pilot analysis on some recessive genes, just to make sure, well, is it actually picking up disease-causing mutations? Does it make sense? Is it of any value? Is compressing the data in this way of any value in the Genomics England data set? And so we published a paper, and anybody who's interested can grab this paper and have a quick look at it. It came out towards the end of last year. And so I'm not going to go into great detail on it, except to let you know we developed the GenePy matrix for just under 3,000 genes at this time. And targeting recessive disease genes. And we did it for the 78,000 individuals in Genomics England who either had rare disease or they may have had cancer, but we did not look at cancer data. We just took their germline data. So it could have been a rare disease patient or their parents or the germline DNA from cancer patients. So we have this massive data set of 78,000 whole exomes, and we created the GenePy scores for these 2,800 recessive genes. And at the time, it was a student project. We arbitrarily asked which individuals are ranked in the top 5 out of 78,000. Are individuals ranked in the top 5 for any of these recessive genes? And we looked, when an individual was ranked in the top 5, we looked at that subset of rare disease patients and asked, well, actually, is there causal, where the diagnosis was known, is there causal variant in that top 5? And for 71% of them, their causal diagnosis was within the top 5. So this was done at scale by one individual, was able to identify 70% of the diagnoses already made over many years by many people, at the touch of a button. So that was very interesting and very useful. But then of more interest was, well, what about those people who had had a negative diagnostic report in the Genomics England research environment? Did GenePy help to unpick perhaps the disease genes behind that was causing their disease? And for those individuals who didn't have a positive diagnosis, we went back and looked, well, were they ranked within the top 5 for any of these genes? And if they were, did their clinical diagnosis match the gene in which they had this high ranking? And if it did, we then looked at the segregation, because of course these were recessive genes. We made sure that if there was parental data, that one variant was coming from mum and one variant was coming from dad. Further details are in the paper, but the bottom line for this was that we did actually identify some hundreds of individuals, about 122 previously unmade diagnoses in these individuals. So in the phenotype match cases, about 20% of them, we had a diagnostic variant that met, or diagnostic variants that met the ACMG guidelines, and we were able to feed that through the diagnostic discovery group, so the data went back to the clinicians and back to the patients as a diagnosis. And in actual fact, another about 50% of those phenotype match cases, we actually do believe are probable diagnoses, but unfortunately we have this problem of variants of uncertain significance, where very often for recessive disease, one of the variants might be well known, but the second variant is a new variant that hasn't been identified before, or for which there is very little functional data proving that it does something in that gene. So it's a highly likely diagnosis. It's predicted to be deleterious, but that second variant does not meet the clinical criteria to go back and say, this is your definitive diagnosis. So more research needs to be done on those. So that was in Genomics England data, and of course that was in rare disease patients. But then we also more recently applied the GenePy algorithm to look at quantitative traits. And this is where we used the 200,000 individuals with the phase 2 whole exome sequencing data from UK Biobank. We created the GenePy matrix for them. So again, 20,000 genes, and this time there's 200,000 individuals with whole exome sequencing data. And for each of those individuals, we took their earliest measured blood cholesterol level. And what we would do then for each gene in the genome is we would take the GenePy scores, and that's what's shown here on the x-axis. So we took the GenePy scores, ranked them, and the individuals with the smallest genetic scores would be in the lowest ranked bins, and we popped them into bins of a percentile bin. So there's 100 bins on this cohort moving along the x-axis. 100 bins on the x-axis, reflecting on the left-hand side the lowest genetic scores or GenePy scores for any individual gene, so scores of 0 probably, and then the highest genetic scores or GenePy scores on the right-hand side. And on the y-axis is the cholesterol, the blood LDL cholesterol level observed in the individuals. So you can see across, if you move across those percentile bins, you will see that across most of the percentiles, it's fairly stable. The average or the mean cholesterol level is stable across that whole cohort. But what we actually did was we took the bottom percentile bin and compared it with the top percentile bin statistically using the Mann-Whitney test. And we asked, is the GenePy score in the bottom percentile bin, is the cholesterol level in those individuals with the lowest percentile different to the cholesterol level in the individuals with the highest percentile? And we did that for all genes in the genome. Now, this graph has the data for the most significant gene when you conduct that test at scale, and it's the PCSK9 gene. Now, PCSK9 is known to be a disease gene for familial hypercholesterolemia. It's a gene that breaks down the LDL cholesterol receptors. So if it's breaking down the LDL cholesterol receptors in your body, it means the cholesterol remains in your blood for longer. So if this gene is very good at breaking those LDL receptors, if you have a gain of function variation in this gene, it can cause familial hypercholesterolemia and the cardiovascular disease that would ensue that. So a gain of function in this gene is associated with disease. But what we observe looking at these data is, in actual fact, the individuals with the highest GenePy score in the PCSK9 gene, so when the gene is broken, when it has a high pathogenic burden, when my natural version of the PCSK9 gene is not functioning well, then my LDL receptors are more persistent on my epithelium and actually remove cholesterol more effectively from my system. And so you have a lower cholesterol level. And this gene and this analysis in UK Biobank, where you're not actually just looking at people with disease, you're looking across a population, it shows the value of actually many of us in the population have a naturally occurring protection against high cholesterol levels because we have lost the function of this PCSK9 gene that breaks the LDL receptor and removes cholesterol from our blood. So that was the story for the top identified gene in that quantitative trait analysis. But of interest, and we did that across the whole genome, and we very strongly identified all the genes that are implicated in the Genomics England panel app for individuals with familial hypercholesterolemia. So it demonstrated the value of applying GenePy at quantitative trait level as well. So I'll move on now to talk a little bit more. So we've got our GenePy matrix. It appears to be picking out rare disease variants quite effectively, and it appears also to be working at a population scale looking at quantitative traits. So we now have that algorithm set aside, and I'm just going to talk about the clinical data and how we use large language AI models to pull and to scrape clinical data to merge with those genetic data. So, and this is work done by some postdocs and students. So I've got Alex and Zach here up in the top right-hand corner. So on average, hospitals produce petabytes of data every day. And it's estimated by the World Economic Forum that 97% of those data are going unused. And I found it frustrating with my IBD cohort because when we started recruitment, we had research nurses writing down pages of information about the individuals on that single day when the patient was recruited. We've evolved over time, and we think ourselves quite sophisticated because now every 3 months we hit a button and we download all the blood test results longitudinally. If there's any changes to the drugs, if there's any surgeries, they're coded and we pull those down. If there's any ICD-10 codes in the data, we pull those as well. And things are much, much better than they used to be. The research nurses are able to recruit patients much more efficiently. But I found it very, very frustrating because some of the richest data for my IBD patients and for many other patients who have imaging data or histopathology data, the really rich information is that unstructured data, those reports following having a biopsy sample taken, and the pathologist writes out a report with lots of rich data in there, and really I can't get my hands on that for research. And the same if there was imaging, be it MRI or ultrasound scans, very difficult to get those specific data back. I have in the past tasked some PhD students to reading reports and pulling out and essentially creating standardized data, but that's inefficient and just not doable at a large scale. And so we have these unstructured data, and we have in the past used the clinical curation team to then create structured medical records that could be useful for scientific research. But at a recent check, when I asked our hospital digital team, how many of these unstructured imaging and histopathology reports do we have for the 1,500 individuals with inflammatory bowel disease in my study? And the answer was in excess of 30,000. Imaging and histopathology reports. And there's no way we can get humans to read those 30,000 reports. And of course, once they would get even a fraction of the way through, there's probably going to be 1,000 more reports ready. So it's just not doable. So we needed a large language model. We needed to replace that manual curation step with a large language model that would pull out these data in a manner that was reliable, standardized, and tuned to work well for inflammatory bowel disease. And so that is what we've done, and we used this methodology. So we had these clinical reports, about 32,000 of them on the left-hand side. We had subsets of those reports that were manually curated by clinical gastroenterologists, and we used multiple individuals to do this manual curation step to make sure that the humans were actually pulling data consistently as well. And then we developed these two schemas, one for the histology reports and one for all the radiology reports. We actually tested a large number of quite a few large language models, only ones that we could download to our local high performance compute. No data were ever exposed to the internet. So what we would do is we would get those 32,000 reports, redact any personal identifiers, NHS numbers, dates of birth, obviously names, postcodes, all of that was gone. And so we would just have the meat of the actual report that the radiologist or pathologist would have written, assigned to the study ID number, and the large language models would read those and create structured findings from those data. And we tested, as I say, multiple large language models. We used standardized ontology, SNOMED, Uberon for regions, CT, NCIT, ICD-10 codes. These were the dictionaries or the ontologies that we were translating these data into. And then, of course, we compared the outcome of what the humans produced and what the large language models produced. And we went through this process, iterative process of improving and engineering the prompts to make the models work better, so that eventually we ended up with our model of choice, the IBD adapted large language model, that we were able to implement these customized schema at scale to all 32,000 reports and generate these findable, accessible, interoperable and reusable data. Just, it might be of interest to some individuals to know that actually Llama turned out to be the best in our hands at this time. The Llama 70, trained on 70 billion bits of data, was optimal working out of the box, but it was possible to train a much smaller model to work almost as well. And that may be a consideration for people who are conducting research, perhaps without quite so much high performance compute at their fingertips. So this is an example of actually what it did. So here's an example of a raw unstructured report on the left-hand side. And as you can see, there's lots of unstructured text there. So the large language model compressed that and would extract the key statements, and then it would correct typos. It would surprise many people how many different imaginative ways there are to misspell the word Crohn's disease. But of course, it's able to deal with that. It's able to correct typos and interpret these. You can engineer it so it sees the context and it corrects these typos, maps to the standardized ontologies according to the prescribed schema. And so what we ended up then with, sorry, I'll just go back there for 1 second. What we ended up with was the sort of phenotypes I'd never been able to access before. So things like stricturing disease. So stricturing disease is when a part of the gut narrows so much that actually the function of the gut is compromised. And if stricturing disease develops in a patient with inflammatory bowel disease, it can indicate the need for surgery to chop that piece of gut out, essentially. So really, really important endpoint, clinical endpoint and phenotype that we had not previously been able to capture from the clinical data. So stricturing disease, was it present? Importantly also, was it absent? And if it was present, what date was it identified? And how long was that post-diagnosis? So all of these data are now suddenly at our fingertips in a way we had never had them previously. We were similarly able to curate things like fistulating disease and granulomatous disease, very important clinical endpoints, but also any other phenotypes that might have been relevant for those patients. And so then, of course, the important thing to do is, well, now you've got the rich clinical phenotyping data, you've got the genetic data, how do you pull those two things together? And that's what this slide is going to talk to. So I'm going to reveal in a second a graph here, and on the on the y-axis where you see GenePy, and this is the min-max score, so it's the score whereby we've sort of compressed all GenePy scores, whether the gene has a small or a high maximum score, they're all viewed on the same level, so they're normalized, if you like. So we take the GenePy matrix that we've created for the 1,500 individuals with inflammatory bowel disease who have had their DNA extracted, their whole exome sequencing done, the GenePy algorithm executed to create a GenePy matrix, and then we are putting these genetic scores, if you like, on the y-axis. And on the x-axis, we, of course, at the same time, in parallel, we have the same individuals. We've taken their unstructured data, put those through the large language model, and we've pulled out these standardized terms in these individuals and mapped them to HPO terms, human phenotype ontologies. So this is the standardized ontology that's probably most often used in genetic data analysis. And then we used another large language model to ask, well, if for this individual, the little cartoon individual up here, this patient, blue patient shown on this slide, if these terms below, you can see sepsis, ileitis, Crohn's disease, systemic infection, if these are the HPO terms observed in this individual, what is the overlap between the terms observed in that individual and the terms observed in known disease genes? And that's what we ask the large language model to create for us. And that's what we present on the x-axis. So those genes where there is a high overlap between what we observe in the HPO terms we observe in the patient and the HPO terms that are expected for a disease gene, they're going to be shifted for the right on that on the x-axis. And so these are the actual data for one individual. This is patient Southampton proband 0285. And again, looking at the y-axis, what we see is that genes that are up at the top of the graph, those are genes with the highest pathogenic burden in this individual patient in proband 285. And then and so that's what's observed on the y-axis. So high up means high genetic load. So any genes that are down here at the 0 level on the y-axis, those are genes for which this individual has no nasty variation observed. And then if you move over to the right-hand side on the x-axis, you see the genes for which this individual has high overlap of their phenotype scores with the known disease genes. So what we actually need to look at is the top section of the graph. And we've actually colored genes here. We've colored genes in red if they are known monogenic IBD genes. And there's about 100 of those identified in a white paper, European white paper. There's about 100 genes that are known to cause basically a monogenic primary immunodeficiency that happens to masquerade. The individuals often get a diagnosis of Crohn's disease or ulcerative colitis, but truly they're not common complex diseases. They are primary immunodeficiencies masquerading as inflammatory bowel diseases. So we colored all those genes red. A wider panel of any other gene identified in primary immunodeficiency, even if it wasn't specific to IBD, we colored those amber. And all the rest of the genome, 20,000 or so genes, we colored those green. And what you can see for this individual is that they have a very high score. If you look at this red dot in the top right quadrant, they have a very high score relative to the other 1,499 individuals in our cohort. They have amongst the highest score observed for this CTLA4 gene. And their phenotype terms, because the score is shifted to the right, it means their phenotype terms also overlap with the CTLA4 gene. And so then you can very quickly digitally scan through these data and pull out where we have hits in this top right quadrant for individuals. And sure enough, it turns out if you go in and look at the variants for this individual, for this gene, they do have a diagnosis. Previously, their diagnosis was Crohn's disease. That is now in the process of being changed to CTLA4 deficiency. So it is a primary immunodeficiency. And the important thing at a clinical translation level is this was an individual, a male child diagnosed about 11 with Crohn's disease. They had some recurrent infection. They were originally put on an expensive monoclonal antibody called infliximab. They're currently been treated with adalimumab, and that remains stable. But this diagnosis of CTLA4 deficiency means that this drug called abatacept actually targets that mechanism, and it should be curative for this individual. The individual probably over the next years may or may not consider having a family, and of course knowing the exact variants means that that are causing the disease means that pre-implantation genetic diagnosis will be available to ensure that they eradicate this chronic disease from the next generation in their family, if that individual so chooses. So there's both a personalized intervention for the individual themselves, but also family planning consequences for the future. This is another individual from our IBD cohort, and this individual, again, this was a patient diagnosed in their mid-30s actually with Crohn's disease, but our data indicate that actually the correct diagnosis is XIAP deficiency, and we again identify it from this high pathogenic load using the GenePy score in the XIAP gene, concurrent with a substantial overlap with the known disease genes for XIAP. We have other examples. So this individual, in actual fact, it's not a gene that's on the monogenic IBD gene list or on the primary immunodeficiency gene list, but actually if you look at the DOC11 data for this individual, we've just we're in the process of getting the wet lab validation back for this, but it's looking like this patient actually has a DOC11 deficiency, which is an auto-inflammatory disease as well, and can present with symptoms very similar to inflammatory bowel disease. So I hope those three little examples now demonstrate to you, well, probably how smug I was going back to the consultant who originally had said to me, well, GWAS are great and you found loads of stuff, but it's not clinically translatable. Some of these individuals are those consultants and patients, and we are able to actually make the data work for these patients now. But I do still feel we're only scratching the tip of the iceberg with respect to the tools and the power of these large language models to get the clinical data that we need to make best use of the genetic data. So my group is amongst many others that are increasingly using these algorithmic tools to try and look at the vast amounts of genomic data we're generating at scale, whole exome, whole genome, and of course we've got whole transcriptome, proteomics, metabolomics data that can all be layered on top of GenePy scores, which of course are scores at the gene level, much more similar to transcriptomic data or proteomic data. And I think, I hope I've conveyed that in actual fact, applying AI to genomic medicine, it depends on having these three things at the same time: the massive genomic data alongside the rich clinical data in an area where you've got enough compute environment. And all of that requires necessary and appropriate governance. And that is no small obstacle a lot of the time. I hope I've conveyed that GenePy actually is an alternative tool. I don't think it's the panacea for everything. I think it's just one tool in the toolbox that allows us a different way of looking at data at scale in populations, but also at an individual level to make diagnoses. And I do feel that AI and large language models really are going to be a game changer in trying to extract the rich data that are trapped inside unstructured data in the electronic health records. And they're going to make a really big difference to us over the next years in medical genomic research. But an integration of those two things at the same time using either high performance compute CPU or GPU systems is going to really change things. I think we're going an era of really rapid development. And I suppose the other message coming back is that not all common complex disease is really as common or complex as we thought before. That in our hands, certainly for a very common disease, we are identifying multiple cases of patients with a rare disease that is masquerading as a heterogeneous common disease. And we think that's likely to be repeating itself across a wider range of diseases beyond just inflammatory bowel disease. So again, I'd just like to acknowledge, obviously, the university, Iridis High Performance Compute. I think I've mentioned I work for NHS Central and South Genomics, and I'm also, with that hat on, part of the NHS Genomic AI Network of Excellence that has been inaugurated in England over the last year. I work closely with Wessex Secure Data Environment, and following that data environment, it's networked with a number of other secure data environments across the rest of the country. I think collaborative use of these models that can lift and shift and make sure that any results we're creating are generalizable to different environments and different patient groups, whether they be in this country or in other countries. That's very important. We have funding from UKRI, and of course we often rely on very large cohorts to demonstrate value and scale, and Genomics England and UK Biobank are two of the ones that we're using most often. Obviously the patients in my own cohorts are highly valuable as well, and of course we're working closely with Amazon Web Services to find the most efficient ways to deploy these algorithms at scale, and the LifeBit team inside Genomics England. So at that point I'll wrap up, and I'd be delighted to take any questions if there are any.
Fascinating overview, Sarah. Thank you. We already have a few questions. GenePy seems to be a really effective tool in picking up true genetic cause of disease. But you mentioned it's just one tool. What other developments are there? What else is down the track, do you think?
Yeah, so, well, even GenePy by itself, I think it has limitations, like a lot of the other tools that are around at the moment. And any tool that's looking at exome data is, by virtue of that, it's looking at short read sequencing data. And there is a massive smog or layer of confusion with short read sequencing data. It means that we can create variant call files, but a lot of the time we don't know how one variant relates to the next variant. In other words, we don't discriminate phase. So one individual may have 3 variants in a given gene. Another individual may have the same 3 variants in an individual gene. But if they're observed as heterozygous in both individuals, sometimes all 3 variants are on the maternal copy in one individual, whereas they could be shared across maternal and paternal copies in the other individuals. So long read sequencing will be the way to overcome this and help us to phase data. So as a tool, long read sequencing is going to unmask that obscurity conferred by not being able to inform phase. You can, if you have parental data, that can be doable, but of course that triples the cost of the sequencing. It means you have to sequence mum, dad, and child. And it also means that if you have a single parent family or orphans or individuals with a late onset disease in their 40s and their parents are just not available, it means you can't phase their data. So long read data is long read sequencing, and the falling costs of that are an important tool in our toolbox that's coming out. But there are very few very large cohorts with long read sequencing data. There's a lot of different decision support software systems coming through, and I think we're going to see a bit of a burst of those over the next few years. And I think those decision support software, lots of them are scraping clinical data from electronic healthcare records. So that's another tool that's coming out as well.
Very good. And there's one more. What represents the biggest hurdle to widespread deployment of tools like these if they are providing ways to increase genetic diagnostic uplift and reduce manual curation burden?
Yeah, so I think probably the governance is one of the biggest problems. It can be, so if I take Genomics England, it's one of the best and most powerful data sets in the world. But in actual fact, the amount of clinical data is somewhat limited to maybe the high-level diagnostic reason for which that person was referred for sequencing. The data are not rich. We don't have 47 different HPO terms assigned to every individual in Genomics England. And the same can be said for UK Biobank. The clinical data there are much, much broader, but often not very, very deep. And getting the governance to have that live, recent, longitudinal, deep clinical data in the same place as the genomic data, I think it is a sticking point, because a lot of these models, they need substantial data to train and test and validate. And it can be hard to get the amount of data at the scale needed to test these, that in a way that is appropriately governed, and that we are allowed to move those data into the secure environments. So that tends to be a frequent sticking point. I was very lucky to be, or I am very lucky to have a decent compute environment locally and research consented patients for which I can extract almost live the longitudinal clinical data, but that doesn't exist at scale across very large patient communities.
Thank you. Floor is open for any further questions. You need to type in the chat. Just give a moment while we're waiting for any last queries. Again, actually have one just received. In terms of identifying misdiagnoses, how might this influence reanalysis protocols in clinical genomics?
Yeah, so that's a really good point. So keeping up with just first-time analysis at the moment, it can be very hard. It's one of the bottlenecks. Actually, we can generate genomic sequencing data really much more quickly than we can manually interpret it. And nationally in England and across many other countries, there is this bottleneck that you can generate the data, but then it takes some individuals to go and look at the variant call files and compare that against the clinical data for that individual. And that's the bottleneck in analysis, even of the first pass of the data. And that means that relooking at old data is certainly put on the back burner a lot of the time when clinical scientist time is at a premium. So I think for this, tools like this are going to be really, really important in maybe making that first pass diagnosis by an algorithm. And so that the clinical scientist, instead of spending maybe 1 hour or 1 and 90 minutes looking at the clinical data and the genomic data for an individual, they get directed to very quickly, for example, the CTLA4 gene, look in this gene for this individual, because that's where there's a really obviously low-hanging fruit. And if instead of 90 minutes, it now takes 9 minutes to do that, that means that the more complex cases, the time becomes available for the clinical scientist to spend on that. And again, there is no human cost to deploying these things at scale repeatedly over time. As for example, maybe the individual was referred at 6 months because they had failing kidneys, but maybe at age 2, they're starting to develop deafness. And now as the clinical data enrich, you do want to reanalyze the data. And of course, you can just set these things to run, and you can set up alerts that, okay, we've got a hit for something in the top right quadrant. Let's go and look at that, and only spend 9 minutes looking at it rather than trawling through large panels of genes. So I do think there is lots of potential. Again, I don't think it's going to be a panacea. It's not going to fix everything for everybody. I haven't even talked about copy number variants or de novo variation here. So it's not a fix for everything, but I do believe it might be a way of radically reducing the amount of time to get to the low-hanging fruit and to digitize those easier analyses for the clinical scientists. So that finite resource of human manual curation and labor and sophisticated analyses that you can't really program for, that actually we deploy that human logic and expertise to those cases and sweep away much more quickly the easy to look at cases and set these algorithms to rerun as the clinical data mature.
So analysis is continuous rather than an event which has to be remembered and scheduled, and fantastic.
Yeah, or set to redeploy as the clinical data evolve over time. So it could be at individual time points, or it could be triggered by, for example, a new diagnosis or some new manifestation of disease in that individual.
Superb. Thank you. I don't see any further questions. We'll give just another minute as we have the time. But appreciate those who ask questions. Creates a really good kind of close to the session, which again, fascinating, and our great appreciation, Professor Ennis, for the overview. Again, this event has been part of a series and part of a program that Diagnostics runs, and it's something which will be available to all attendees on demand if they want to listen back or consult any further elements of it in the future. And as I mentioned at the beginning, DXRX network is our hub for all of this. So do feel free to come visit us there for more like this, and indeed any further information on this particular topic. If there are no final questions, I'll once again thank Sarah for her time and insight, and to all our attendees, to LabRoots, and to the team who helped put together the event today.
Lovely. Thank you very much.
Thank you all.